Good morning, good evening. Thank you for listening. This is a show about agriculture, horticulture and gardening, focusing on plant science, soil science and microbiology. And today is about the microbes in agriculture. So if you're already watching this back, fast forward to see some slides. I didn't mention too, this information is for educational and scientific purposes, and any of the websites or papers discussed will be put in the description below. And now, fast forward, that's good. You can, and I'm going to give it another 70 seconds for the chat to arrive. So fast forward if you're watching that. $60 seconds. $40 seconds to go till I start. $30 seconds. I'm getting my heart. Five seconds, and I'm going to start. Left off last week at Bio-remediation. I pulled up the screen already. Bio-remediation. So I went through them. So this one was where I stopped. So yeah, why do you want bioremediation? Well, microbes can remediate the soil contaminated with pollutants and chemicals, contributing to soil health restoration. So that can even happen anywhere. Even if you've got good soil, you can still remediate and bio-augmentate, which are the three types there. The middle one, that's when you introduce microbes. The bottom one, biostimulation, is where you stimulate the environment to enhance what you've already got, to break it all down, and then naturally it does it as the third one. It's biogeochemical cycling of nutrients. Involves the recycling of essential elements. Bio means living, geo means earth, chemical means, well, nutrients. So all of those things cycling together. And the microbes, they're the crucial role because they do all the breaking down and transfer, recycling of the nutrients. So that's, yeah. And on the little nutrient cycle, what's that showing? Water and air, penetrate, okay, just a very, very easy nutrient cycle. Soil microbes in nutrient cycling and soil fertility. Sustainable agriculture productivity depends on soil health and fertility, significantly influenced by soil microbes. The microbes are crucial for the cycling of essential nutrients. And there's three cycles they're going to go through: nitrogen cycle, phosphorus and sulfur. Nitrogen cycle, well, I'm not going to go through them all because elements, they change from this back to that. Like azotobacter. Azo, what's the one that does nitrosomus? Converts NH3 to nitrite, and then azotobacter converts nitrite to nitrate as an example. So that's what the microbes help in the cycling. Without that, it's chemically done. That's why microbes play a huge role in these. Phosphorus recycling. So you get a few different types of microbes involved in phosphorus, because they can be phosphorus solubilizing bacteria, they can be phosphorus releasing bacteria. There's a few different classifications in the phosphorus of the different microbes that are involved in the phosphorus cycle. It says mineralization, solubilization, and immobilization. Okay, 3 ways to but alternate through the cycles. And then the sulfur. They're all just as important as each other. Various biogeochemical cycles will be discussed. Okay, microbes as bio-fertilizer. Now this is the rad bit. See if anyone's arrived. No. That's wrong. Microbes as biofertilizer is the go. You can get free, like azotobacter pulls uses N2 out of the air and converts it into usable form nitrate. It's azotobacter. Azospirillum, sorry, does that. Not azotobacter. Azospirillum, yes. And yeah, that'll keep going. I like to keep it truthful. I don't like to pretend or whatever. So biofertilizers or microbial inoculants are microbes that enhance plant growth and yield by colonizing the rhizosphere and plant roots. They also improve soil fertility. That's what I was just about to say. Common biofertilizers include nitrogen fixing, phosphate solubilizing, and fungi. And let's have a look at that. So there's a few different types. Anabana, Azola, that's spirulina. Pseudomonas forans, I use that one as well. Azospirillum, Rhizobium. What's that? Rhizotinobia solani. Oh. So the benefits of biofertilizer is everything. Low cost, easy to use, they recycle themselves, they keep producing themselves under the right conditions, enhance nutrient availability, everything. Reduce stress, get better plant expression. It's really outstanding. The benefits that I've seen from using them are fascinating. Here's some different classes of them. So the fungi down the bottom, phosphorus, there's a few, trichoderma, mycorrhiza, photoautotrophs like cyanobacteria, heterotrophs, different types of food sources. And on the end, you can see different types over here. Some azos, Clostridium, Anabana, nostrate biota there. This is a good chart. This shows you, well, it's a classification of biofertilizers, but it shows you different examples of some species, and genus. Yeah, sorry, there's circulants there. So that's the species and the genus. So if you want to source some of them to do specific things, because my role really, is to get different microbes to do different things. So if I'm thinking, all right, I've got a nitrogen deficiency, well, I can go add some nitrogen fixing bacteria, or I can go and enrich the soil with some azos to produce nitrogen from the air. So by knowing these things, you can get different microbes to work for you. It's kind of like giving it fertilizer, but it's through the bio. So that's why bio-fertilizer. So here's what their abilities are. Well, nitrogen ones produce nitrogen. Without reading all that, that's what they do. It goes into a few different regions of ones that are symbiotic and ones that are free living. Like Azocochrocum, I use that one. And that's the one that doesn't need anything to function. Where at the top, Rhizobia needs legumes to work with. So if you get some Azotobacter, it's sweet as. You just go and put it in your fields. Even if there's no plants, it'll still function and produce nitrogen. And then cyanobacteria, I use that one too. There's a few different types you can get that produce indole cissic acid. So if you want to go, I'm not going into that bunch actually. Phosphate mobilizing. So see the other one was solubilizing. This one's mobilizing. So it just makes the element mobile. So it can be readily available. So instead of solubilizing, it's readily right there. Phosphate mobilizing bacteria may directly mobilize both phosphorus indirectly, and it also occurs through organic acid chelation and inorganic acid production. Very good. Proton exclusion, exopolysaccharide production, cytophores, and enzymatic action. Potassium solubilizing and potassium mobilizing biofertilizers. Some examples are like Bacillus and Aspergillus niger. There's also, what do I use? Mucilaginosis comes off the top of my head from that one, potassium. And might I add too, just by getting all these and throwing them together, that's what companies have done, and they expect it to work. It doesn't work like that. It's some don't function next to each other. They don't like each other. So you've got to be careful. Just by adding and adding, you can't just keep adding and thinking everything's going to be good. Fungi, solubilized mechanism, bacteria. So this is to do with potassium, and it's just showing bacteria and fungi as they're breaking it down. So different things they do, mechanisms and stress tolerances, plant growth promoting rhizobacteria attributes. Potassium solubilizing bacteria has many different acidic benefits. Different organic acids over here. You can see there's heaps of them. At least a dozen. I'm not going to read all of them. Is humic there? No. Fulvic? No. I've got formic. I wonder if it answered like that. There's another one. These are organic acids. They can release potassium ions from potassium-bearing minerals by chelating them. Zinc solubilizing microbes, like Bacillus species, and improve the photosynthetic efficiency of plants like B. junicea. Efficiency of plants. Bacillus, okay. So they've got companies now that have gone and got all these microbes and put them into one, and they're trying to produce the NPK equivalent. Mechanism of zinc solubilization. Show it on that slide. Arbuscular mycorrhizae as biofertilizers. So arbuscular mycorrhizae are beneficial bacteria that colonize in the roots. Yeah, self-fertility help. I'm trying to watch my voice here. Vascular colonization has positive effects. It sure does. Because what they do, they put out branches of hyphae and grow way faster than the roots. And then that hyphae can go and pull in minerals, and it transfers those minerals along through its network, mycelium, the fungi, and then it can make it available to the plant's roots. So that's why they have a really good benefit. So they found to work out quite well. So if you've use herbicides, pesticides, that type of stuff, you might have killed all these microbes. I promote organic practices. Plant defensive signaling through mycorrhizal. No, I'm not going to go into that. Microbes help with signaling and through stresses, abiotic and biotic stresses. Endophytic fungi, bio-fertilizers. So that endo means inside. So the plants live inside the cells, which live within plant tissue without causing harm. And they're the ones that will more than likely get transferred through with seeds. Like Piriforma spora indica is an example. Most endophytes, they have a direct effect, indirect effects usually. They can sit there and do good stuff, or they can have a direct effect by interacting with the plant cells, nutrient systems, and the phytohormones. Clavi, Civi, Pitta, Ceus, endophytes in agriculture. So that's the different species that are used for biocontrol in pastoral grass. Because this pest tolerance in tall fescue by producing alkaloids that deter herbivores. So ryegrass varieties with this microbe in it exhibit reduced damage from insects in New Zealand and Australia. However, their application has been largely restricted to grasses, limiting broader use in other crops. Wow. Endophytic fungi and cold stress resistance. As you've seen before, it helps with cold stress. They put out enzymes that really work really well. Oh, that's this one. This is other types of psychotrophic microbes, which are cold-tolerant microbes working below 10 Celsius or 50 F. This is what they do, because it's one of my areas of interest as well. Because why should we not grow in winter? There is microbes that do the same things at different temperature levels. Why don't we just get the microbes at the temperature level where the soil is, and we can continue the growth and development instead of it going into a kind of a dormancy phase. Interesting, isn't it, this plant science stuff? Must be into another section, bio-pesticides. See if there's any questions. No. There's Raptor. There you go, Raptor. Okay, mate. Hello, mainly mushrooms. Going well. Yeah, doing microbes for a few weeks. This is part of a course that was written last year, so the information is quite up to date. I got 85% in the exam. Oops. Getting all flustered because bio-pesticides derived from natural substances, including plants, microbes, and nanoparticles of biological origin. Bio-pesticides, yes. It's a comparison here with synthetics and bio-pesticides. Well, the difference is synthetics kill everything. They target everything, your beneficials and your bad, where bio-pesticides pretty much target only the bad because they're living things. Mind you, pyrethrum isn't, and that will target good ones. But in general, and biopesticides, if you use microbes or biological insects, they will build up in your garden, in your compost heaps, and you can really sell your soil. Classifications of biopesticides. Natural enemies, plant incorporated products, biochemical and microbial pesticides. Here's some fungi, Bio-Bassiana and Metahizium anisople. They both produce muscadine disease. So if you see here we go. Yeah, this one's green. I use both of them. Metahizium puts out a green muscadine disease. The hyphae is green, and Bio-Bassiana is white. So after it infects the insect or whatever, you'll see them looking furry, and you can tell from the colour which fungi is growing, which beneficial fungi is growing. BT, Bacillus thuringiensis is rad. It targets so much. Viral pathogens. Stenonema. There's beneficial nematodes too. Stenonema felti is one that's for fungus gnats, and oh mate, does it work well. It just blimmin' works. Stenonema felti. If you've got fungus gnats, don't muck around with anything else. Get that one biopesticide. It will target the one thing and fix the whole thing. That's an example of how to use microbes to your advantage. Everything else stays in check. Natural enemies are organisms that naturally control pest populations. So if you keep spraying and spraying your bad pesticides bought from the shop that are killing everything, all the natural enemies will die too. You've just knocked out the whole cycle. That's why a lot of gardeners struggle, because they haven't got a good biosphere intact in place. Plants incorporated protection are pesticides, substances produced by plants as a result from genetic engineering. Okay. Plant incorporated protections like Bacillus thuringiensis, crops. That would be BT cotton, where they went and, there is down the bottom. The BT cotton. Oh, it says cotton. Well, they got the bit out of that causes the problem and genetically put it into cotton, and now cotton grows, producing that same effect that was in Bacillus thuringiensis. So it has the toxic protein. There it is down the bottom. So it produces that now cotton, the same thing that BT did. Mode of action. No, don't need to know the mode of action. I'm not getting that far into it. Biochemical metabolites. So biochemical pesticides, biopesticides are naturally occurring substances or synthetic analogues that control pests through non-toxic, like neem, garlic, pyrethrum. So but be careful with these ones because they do get rid of some beneficials too. That's the only bad thing. Secondary metabolites produced by Bacillus thuringiensis, and wild mushrooms, strobilurium, biochemical botanicals. So botanical extracts are plant-derived substances used as biopesticides to manage pests, insects, weeds, fungi, bacteria, everything. And some botanicals are neem. Neem's pretty good. You can also, once you get the neem plant and you put it through your cold press, get the leaves or the seeds, it has the highest azadirachtin content in it. You'll put it through your cold press. That neem cake, which is the results after it, has a NPK higher than chook poo, or equal to chook poo. It's a good plant to muck around with. Pyrethrum. They make a synthetic version too. Be careful at the shops if you think it's pyrethrum. It's probably not from chrysanthemum. It might be a synthetic version, which is bad. It takes ages to break down. This one breaks down really fast because it's natural. Garlic, capsicum. You'll see a few botanical, can't remember the name of it now, but they've got it in with those, put in onion, garlic, capsicum, different things like that. Pepper, oh, that's capsaicin, that's what that is. Quassia, must be something. Biobotanical, here's secondary metabolites. They serve as essential biopesticides derived from plant and other sources. The compounds play crucial roles in pest management due to their diverse modes of action in targeting various pest species. And semiochemicals, here's the sweetness. Modes of action of these: they disrupt mating cycles, they attract insect traps like pheromone traps, and they naturally repel insects. So that's all of the, I suppose you can say, terpenes that come out of them. But the pheromone traps are quite well, because in mating season, that's what the insect or when they're going to reproduce, they'll be very interested in that pheromone, the opposite. So Bacillus thuringiensis is a type of bacteria that produces special substances to fight against pests. Okay, works in, it's very wide. I use that one. It's very wide. It also puts out indole acetic acid, which is a auxin phytohormone. Bacteriocins. I'm not going to read all that. BT produces several bacteriocins, so all these that are beneficial. We're all different types, but secondary metabolite spinosins. Spinosad. It's a really, really excellent way to get rid of thrips. It's very hard to produce things to get rid of thrips or worms, where those two things just eat it up. And spinosad comes from the gut of Saccharopolyspora spinosa. So the bacteria has this little chemical inside of its gut, and they extract that through secondary metabolites processes in batch culturing and derive this spinosad. And when you get spinosad on the side of the bottle, it'll say the dilution rate, and it's really low. It's very, very effective, and it's organic as well. Really, really good. I've used this a few times. Mode of action. There's a mode of action for you if you want to read it. Targets pests. Yeah, look at that, worms. It just smashes the worms and thrips. Amazing, it works well with thrips and worms. You spray it on it, and it's like an instant thing. Anyway, what about it? And here's the other strobilurium from the wild mushroom, secondary metabolites from that. It represents a new generation of fungicides. It's isolated from the wild mushroom. It inhibits mitochondrial respiration in fungi, disrupting energy production. Pretty cool. And again, this is organic too. Down the bottom, studies on pearl millet downy mildew showed it provided up to 91% protection. So this is all organics. You can get really good results from organics. Production and formulations of biopesticides from secondary metabolites. It's in the lab where they culture it up and extract it. That's the easiest way to say it. They start with the culture, grow it, they'll stay at stationary phase is when the bacteria puts out the most secondary metabolites. So they'll extract it then and then separate. So microbial biopesticides. It's a class of microbial pesticides. Microbial biopesticides, that's what I've been just going through. It says bacteria, fungi, viruses, and nematodes. That was what I thought we were going through. Classifications. Endospore forming, non-endospore. The best thing about the endospore, say, is if you've got a Pseudomonas or a Bacillus, genus. They look like a shape of rice or a tic tac. And then once it goes into the stationary growth phase, the third phase, it starts to produce the endospore because it thinks it's going to die soon. And one half of it will have a round little spore-looking thing into it, or a cyst in fungi, and that's its reproduction bit. So once the cell dies, it's got a little bit in there. Once it gets hydrated again in the right temperature, it'll regerminate and start again. So that's the best bit about using these biopesticides. With their spore forming, that's how they can keep reproducing. And then they'll double, so you get double the amount. They'll reproduce in summertime, spread more. Before you know it, you've got the best garden in town. You're not doing nothing. Organics rules. What's this? Bacillus who? Populate. Oh, see, new things all the time for me. Obligate anaerobe. Obligate. Microbial biopesticides, entomopathogenic fungi. Well, they were the ones that has the Bavaria bassiana, Metahizium anisople, and Trichoderma. And here's over here, it shows the muscadine disease. There's the white one. So it's, yes, Bavaria bassiana. And the green one is the Metahizium. See how it sort of looks, there's the bug, its antennae. See how it's furry, fungi? And then the last one, Trichoderma. Trichoderma is rad. It's a fungi too, beneficial fungi. And I can say that I use Hosarium, and it's the only one of the Trichodermas. Use variety or get a few other different ones of Trichoderma because they produce IAA. This one doesn't. Microbial biopesticides. Spores infect insects. That's what they do. Their mode of action is they'll plant their spore inside of the insect, and then that spore grows, and that's what you see on the outside. So when that dies, you've got heaps of Bavaria bassiana spores sitting there. Righto. Organics. Microbial biopesticides. Oh, here's how they work. Just by, see, they put their hyphae there and they germinate, go inside and start germinating inside and take over. Entomopathogenic viruses. It's another way of attacking a crop. If you've got a very expensive crop, this is an option that the horticulturist can take. Examples of viruses to beat different insects. So you might go, if I order SE NPV, because I got beat armyworm, I know that will work. So that's an example of how to do it. It's just that easy. Microbial. And I say that easy, it's not that easy, but by having these tools in your kit, it makes it easy. And bringing you success through organic practices. Microbial biopesticides. I use the more modes of action. You can read that while I get another little lolly. Next. The last section, nematodes. Entomopathogenic nematodes, like Stenoneum felti, fungus gnats. Yep. Nematode mode of action. Some examples down the bottom. Way you find out how to get what you want is put in the search engine biological pesticide for fungus gnats as an example, and then it'll come up Stenonemathalti. Then you can put Stenonemathalti into search to buy that, and then see who sells it, and then buy it. Protozoa, single-celled organisms that use quite well as well. You get beneficial ones. Haven't had much experience with it. There's some different types they do. And they infect the insects, their mode of action. Bio-pesticides from microalgae. Huh. Some types of microalgae produce biologically active compounds, which can fight off bacteria and harmful and fungi. I'll use this one. Platensis, Atherospira platensis. There's Anabana and Nostoc. The other two types of cyanobacteria. I've had the same one, same culture for, 5 years. Organic culture too. And it's, all right. So types of microalgae. Just protect plants. Limitations of biopesticides. There are actually negatives because there's storage problems. How do you store the blooming things? Most of them, like even leachette, worm casting tea, there's a shelf life of 3 days because it'll turn anaerobic. All the oxygen that's been in there for the oxygen microbes, they will have used all that up. Bang, it's going to turn anaerobic, producing more anaerobes. As an example, specific, different culturing, like spinosad. Yep, hard to get amounts. Let's go to pest management. Is this onto a new section? Yes. IPM. I think it's the last bit. Yeah. Let's buy a pesticide. I'll stop there. Oh, yeah, 40 minutes. Raptor says just plants transplanted and used some mycorrhizae. Good refresher. Hard to imagine developing a resistance to mitochondrial respiration. Yeah, now you know. You can. You produce different, that's when the plant starts to show adaptations. So I've seen some in my plant science with my fiddling around. If you go and, actually you don't watch it, actually Raptor, you might be interested in, yeah, Raptor, you're still watching. In this channel, it's my other channel, and I do a lot of plant science because this, as you know from people, so yeah, I go and started another one. And what were we just talking about? You were talking about you started something. Microbiology. I'll show you. Here's a link of my other one because I'm going to delete it. I'm not really promoting it from this one, but for those who can I revoke that? I can delete it. That YouTube there is my other one. And I'm going to delete it because I'm not really promoting it from here. you just might be interested because I upload videos every 3 days. And some of the plant science stuff's fascinating because all my experiments are working. All this knowledge that I've got from these studies, everything's working. And I'm getting expressions that are amazing. Like, what's an example? All right. It's a world first, and I've produced seeds that produce broccoli and Brussels sprout on the one stem out of one seed. And I got a 30% success rate from putting 10 in. Thirty 30% success rate, yeah. And I put it into the search engine and it says it can't be done. Well, I got 3 plants that have broccoli up the top, one big head of broccoli up top, and they got Brussels sprouts coming out of the nodes down the sides. And that's one example. I've got a few other things that are cool too. But Raptor says, by the way, you've convinced me to give the neem seeds another go. Good. Yep. Just start playing around, and even if they fail, I've got so many failures that have happened with my plant science stuff that's not funny. But I've learned, because a failure isn't a demise. It's a way of learning. I won't do that again. Oh, maybe I could have done this better. And all of a sudden it works really rad. Sowing the neem seeds tonight at 25. Well done, Raptor. That's cool. They're massive too, aren't they? Those seeds. I hope they're not dormant. And you know how to do it. That's the way. If it was me, I'd put some water out in a cup, put the seeds in, let them float. In 2 or 3 days, some of them should have cracked, and I'll transfer them into a different media then. And if they haven't, I'll look at dormancy techniques. I'd also be putting them in with an apple into a bag, because that's ethylene will help stimulate the germination process. Yeah, the size of kidney beans, he says. Raptor says, massive. That's cool. That's the way. I'm envious of you. I've just had a couple of cuttings from my hedges that I'm multiplying. And it's very cold weather at the moment here, so I'm happy for that to happen. Raptor says, apple is a great call. Okay. Oh, it's a great call. Oh, yeah. Yeah, it's, and make sure you wash the apple first, because it's got different fungi on the outside, and some that's maybe growing inside, it might come to the outside of it and contaminate the thing. So I just wash my apple. I get one apple, it lasts 3 months. Different species don't last me as long for my dormancy technique breaking. So I've got these pink ladies that seem to last 3 months in the fridge. So every time if I'm going to germinate seeds, I'll put some seeds in a little cup, float them, and put the apple in the little fruit bag, and then seal it up and look at it in 2 or 3 days. And remember, if you get too much ethylene, you get the tripling effect. It's the short hypocotyl, and it's got a J hook as a root, and it's for the hypocotyl stem bit is very fat. It's not bad, but no, we're not talking about it today. Right, it just says also got some moringa seeds with the order. Oh, that's all right. That's moringa. How can that be beneficial? Oh, it's used in traditional herbal medicine. As I'm reading here, I can show you. There was I've got another course that I'm doing, an Indian medicinal herbal course that I'm doing, and it's on different herbs like this, moringa. And one example was what's that wort? St. John's wort. I got some of that growing, and it's bad for animals. So these might be beneficial in one way, but just look to make sure they're not, like, know everything about it before getting into it, because you're becoming a doctor. Raptor's pretty good because he does that, but I'm just saying for others. So that liver injury, see? So you can get toxicity from using moringa by using too much of it in the liver toxicity. So that's, wow, that's pretty, that come up at the right time, didn't it? Yeah, because they're herbs. You know, we can even die from like oxygen and water. So everything's toxic at different amounts. So I'll probably put that in there. All right, it says, I buy pink ladies for my family. Going to buy a tree in winter. No, don't buy a tree. Grab the pink lady seeds. Try and start it from the seed. I have, and it's hard. That's what I like doing too. You like doing that stuff, Raptor. And some of the pink ladies, you'll find after they've, you'll pull them, like after mine go age and I've got to throw them out in the compost heap, I'll cut them in half, and you look at the seeds and they've germinated inside, and then they've got the tripling effect because they got too much ethylene. So the root will be too short and J-hooked and a bit brown. It's pretty funny. The microbes, we're probably halfway through the microbe course, at least, the bit that I was going to talk about. Is there any suggestions after that, what I could talk about? Please put them in chat now, because I'd love to hear your thoughts. What else is there? I'll wait till the next slides for next week, because I think we're up to the last little bit. Let's have a look. Last week might be the next show. I skimmed a little bit today because I was worried about my voice. Maybe up to this one. Hey, look, I double clicked and it came up. I don't think we did. Pest management. No, we haven't been up to that one yet. Oh, so that's halfway. Okay, good. So I've got a couple to go after this. That's good. Good to know. So I've got a bit of information, a few more weeks of content. It's hard to know what to chat about. My subjects next semester aren't overly overly exciting, but they're good for me. I'm happy. That university sent me a T-shirt because I got I won some awards at the university there. My pictures on their website for my good results. Very happy about that. Every time, Raptor says, every time I cut mine, they're germinated. I'll put them in some potting mix. Oh, every time you cut the pink ladies, they've germinated too. That's from the ethylene, because that's how well it works, mate. It's because remember with the seed germination, it was gibberellic acid and ethylene are the two phytohormones that a seed needs to produce its radicle and to start its growth process outside of its shell. So yeah, I hope you have luck with your pink ladies. And then you can even, you don't have to use the whole pink lady. Did you know that your ethylene production off gas, VOC it's called, from the apple, if you bruise or cut the fruit, it increases a lot more. So you can even cut the apple if you want, but I don't, because the apple goes too brown, so I just leave it as whole. But if you want to use maybe the seeds, you can do it this time. Just something different. Another thing too, Raptor, you can get into, and everyone listening, is they're called fruit salad trees, fruit salad plants, where they've got multiple species growing on the same stem. It's kind of like I did with my seeds, but you use through cuttings. So you might put an apple and an orange plant, you'll grow them both side by side, separate pots, and then start taking cuttings off one and grow into different ones. So you'll produce the same stem with 2 different species from it, and they're called fruit salad. And they sell for at least $100 for one of those trees in Australia. So it's handy having a greenhouse and doing all these little experiments with your plant science, because you can make massive money with it. Get into the rare and weird types, and you can get some good action. Your time will be very valuable. That's the bit I like about it. Even I've got to keep on topic, I suppose. Microbes. Yeah, going a bit off topic about plant science. Right, just says, yes, I've wanted to grafting with apples and prunes. Oh, yeah. Sweet. It's kind of surprisingly straightforward. It's you do it quick, because now knowing about microbiology, that's anything will want to get into a cut wound, and if it's wet, bang, those spores have just germinated. So if you graft in relatively sterile conditions, you'll have high success rate, because it's kind of tissue culturing. People do grafting outdoors or in big windy areas, you'll see they'll probably have low success rate, or they'll have hardy genetics. But yeah, that's all these microbe courses that I've done. They've all collated to be so relevant with the plant science. It's fantastic. Today my voice held up to an hour. This is good. Put your questions in chat now, please, because I'll be finishing the stream soon. Next week we'll be again continuing about IPM measures, different ways to guarantee you're going to have all your plants will be successful every single year just by IPM. Writing it on the calendar, what dates to get what dates to do different tests, and you can have it all dialed in. So it's going to take care of itself. And I look forward to buying more microbes and beneficial entomopathogenic fungi and bio-pesticides. That more that I can get on the property will only help me down the track. As long as I can keep all the pesticides from the neighbours at bay. So I'm going to grow all the hedges. That's why I've been multiplying all the hedges, and it's been doing pretty well. Raptor says, I've been asked if I regret doing all of the chem and microbio now that I'm a horticulturalist. Not at all, says Raptor. Holy mackerel. People that have asked if they regret doing all the chemical and microbio, they're the people that are into their into their synthetic feeds, because it's too much education for them. They think, oh, how do I learn about that? Well, it all falls into place. Don't expect to be a doctor on the same day. You've got to learn it. So just stick with it. Like that, anybody interested, hit me up if you want to do, they've got this organic certification course, the one that I went through last year maybe, that's open now for remote learning. So you don't have to be in semester or anything like that. You can do it, and it's going to cost you $40 US or $50 Australian to sit the exam. Goes for 12 weeks, but it's a self-paced one. So if you're interested, I shared that with someone else in Weed Nerds the other day. But yeah, because this study, it pays off massively. I've got many years to go of study. All these different new scientific things that they're discovering. Like in winter, people haven't even worried about growing in winter at all. Well, they're pretty much dormant. Well, get some psychotrophic microbes which produce nitrogen and can assimilate it quite well, and get the crops to be modified a smidgen so that they can grow in colder temperatures with your microbes. Bang, you've got success all year round. you've got a cold winter variety of any crop. So that's why I'm really invested my time into the psychotrophic microbes, which are microbes that can do their growth and development around freezing level. It's just one example. No one's done it yet, or very little people have done it. So there's so much potential for success in your own field through these areas. I love it. Thanks for everybody for listening and for helping. I appreciate people that give me their time and like the show. Press the like to if you liked it, please. I'd like to get more. How you can help me is try and get more audience in, try and get some more people so we can get more interaction, because I enjoy the questions and helping that way. So that's how you can help me. And for those people, members and subscribe, thank you very much for that. It's appreciated, because I don't get anything out of this. I just like to help others, because I'm doing extremely well with my plant science, soil science, and microbiology, and I'd like others to get the benefits out of it too while I still do these streams. Because soon I'll have too much time. I won't have enough time, I reckon, in a few years with all my other outside gardening that I'm doing. Everything's working. I've got a garlic farm, and it's just working well. Every year I'll be multiplying the seed. So next year more, better seed, more resistance. And before you know it, there'll be too much for me to do, and I don't know how I'll go for time-wise. So, and then I'd like to get into some other crops. I'm doing kiwi fruit too. They're worth a couple of bucks each, just for one. And I've got a good dozen plants growing, at least a dozen trees of a few different species for kiwi fruit. Be good to cross them, make new varieties with different tolerances with these different microbes. We can get them growing more actively in winter with some psychotrophic microbes. Right, so congratulations on the awards, Aussie. Have a great weekend, everyone. Thanks, Raptor. Appreciate your nice words, mate. Yeah, GMF, you've been baking, mate. G'day. But you're here, he says. That's good. No worries. Still got more microbes next week. More IPM strategies to maintain your crops all year round. We can do plant science all year round. How cool. Congrats, Aussie. Oh, thanks, mate. I just got some good results from the university from my last semester's studies, and they gave me a t-shirt as an award, and I got my photo on their website. Only me and 4 other students out of the many hundreds of thousands that study at the unis got the results. So I'm Rap, so thank you for the nice words, people in chat. Raptor says, try and grow golden kiwi fruit. I'm growing them, mate. Highly prized and expensive in the US. It's a New Zealand produce. I've got, I've got 5 greens, hang on, and I've got 12 goldens at the moment. Oh, and I've got some actually outside too, but no goldens, I've only got greens outside. But yeah, the goldens are, Mum likes them. I don't like them as much. I like the greens that are a little bit more tartar. The goldens are a little bit more sort of sweeter, and I like the tart bit. But I did the goldens actually for Mum. That's why I grew those. And if you want to grow them too, don't buy them. Just grow them from seed. I grew mine from seed. So on that channel, my other channel, I made a video on how to produce kiwi fruit from seed, and that's how I've done it. I've done it a few times now. Just it's all about sterility, basically, and get good genetics. Don't get ones that are gonna, pick your fruit nicely, ones that are gonna age well, and all that sort of stuff. You enjoy green grapes, yeah. Enjoy tart like green grapes, yes. It's good. Green grapes, eh? They'd be good to grow too. Make some wine out of it. Everyone grows grapes. But yeah, the kiwi fruit. Been thinking about climate change and adaptation for the future, because crops are going to go in and out. Species are going to be too hard for people to grow because it's going to be too hot. They're going to get different plagues of grasshoppers. So try and consider that with your future growing techniques with your crops. because there is massive money to be made by you listening by yourself if you choose the right crop with the right genetics. And you can start it all from home by seed and just keep multiplying by that. I've got, say, an example, those kiwi fruit. I've got 20 kiwi fruit trees. You're supposed to space them 20 feet apart, 6 meters. And then they vine up. By the third year, they'll grow and they'll be touching. So that's just one example where you can do everything yourself with a little bit of space. Look out for people that live close. Maybe they're not using their land. Maybe the person's too old. Maybe you can help remediate their land and give them some free produce at the same time. There's different ways where if you live with constraints where you live, you can always maybe try and get help from someone else with a good bargaining technique. No money, don't exchange money, because with crops fail, you'll be stuffed. And a lot of plant science stuff, there is failures. There's times when crops don't perform the way you want them to perform. So by having those fallbacks where you're not heavily invested money-wise into it, it makes it easier for success. Because you don't borrow money for starters. Like, for instance, garlic. I just went and turned $400 worth of seed into 4,000, and the next year it'll be into at a zero, and the next year it'll be at a zero. You can do everything yourself with the right niche market in your climate conditions. So see what sort, you have to get the temperatures, the ground temperatures of where you grow at the winter and summer, and see what pests are around, and see what's most expensive in the store to buy, and choose. And if you choose something like garlic, it's very pest resistant. We're going through all these IPM measures. There's very little you've got to do with that, because it's used as an actual it's as a botanical you saw today. So you might want to get into growing chrysanthemums, or growing, not chrysanthemums, but garlic or onion or stuff like that. I'm rambling a bit. But yeah, thanks for everybody contributing, listening. I really appreciate it. I think today was a good stream again. I hope some people got some stuff out of it. And I'll be doing the same thing next week with the microbes. Probably halfway through that course that was written last year. So it's good information. So I hope you can join me next week, same time. About an hour before, actually, it's not the same time. Sorry. Thank you, everybody, again. Have a nice week. Drop some questions if you want. I love your questions. And press the like if you like it. Happy breeding, happy growing, good health to you all. Thank you, and bye-bye.